And I will share my screen.
Vivian, I started recording and it's showing that you're recording as well. And then do you see it recording?
Yes, perfect.
Perfect. Okay, so Vivian, you can go ahead and show your screen now. We will not use, it looks like we only have about 20 students logged in today. More will probably log on, but that's a sufficiently small number that, you know, if students have questions, they can either save them till the end, or they can, you know, feel free to go ahead and unmute. So don't put them in the chat box because I will not be monitoring the chat box for these lectures, okay? So if you have a question, unmute at a convenient time and ask Dr. Bellafato directly. And at the end of her lecture today, Dr. Bellafato does have some things that she wants to show you. She has some different parasites, some interesting parasites that many of you might be interested in taking a look on. So I encourage you all to stay on at the end of the lecture for that demonstration. All right, and I will turn it over to you, Dr. Bellafato.
Thank you. And now I'll do a big screen. Can I do that? Okay, can everyone see what they need to see?
Yes, the screen looks fine and the volume and the audio is fine as well.
And do I want to hide the thumbnails? I'm just thinking. No, I should probably keep the thumbnails of it. Now I sound like all these other people when they're doing talks. If you have so I'm going to turn off my hide thumbnail video, okay? If you have a question, please just speak up. Can you hear me?
Yeah, we can hear you.
And you can see me, right? I guess hiding thumbnail videos didn't do me any good. There it is. Okay, if you have a question, please just speak up. So welcome. This is, of course, the most exciting lecture because it's parasites. And I'll tell you what parasites are. And I'll tell you about parasitic diseases that infect people. And that's important because as you know, you finish your master's degree, you guys will be considered to be the experts in your family and among your friends of all things medical, right? So you need to know about parasites, partly for that reason, and partly because they are a big problem in this world. Many, many people are infected with parasitic organisms. It's what makes people do poorly in school, do poorly in the field. It's what makes animals sometimes not be very healthy. And so parasitic diseases are really a big influence on our social economic health in our world. So the first slide is sort of cute. It says, welcome back to What's My Intestinal Parasite? And the purpose of this slide, can you see my pointer?
Yes, yes, I can see the pointer.
Okay, I don't know how to change it to a different shape. Is that okay? You can see it?
Yes, it's fine.
Okay, so this is simply a cartoon demonstrating that there's lots and lots of different types of parasites, and figuring out which one you have is, or your best friend has, is something that we'll discuss as part of our lecture. Okay. So first of all, what parasites are. You might think of a virus as being a parasite. It is the ultimate parasite, isn't it? Because it lives off of us. However, we use the word in biomedicine and biology for parasites, meaning eukaryotic organisms. So those are those with cell nuclei, right? They're not bacteria and they're not viruses. But they come in two flavors, if you will. They come as single cell or multi-cell. So what's really great about having a lecture on parasitology is it makes you think about single cell organisms like amoeba and paramecium, and multi-cell organisms sort of like us and worms. So it's a good way of sort of, you know, a 1,000-foot view of biology. There's two types, as I said. There's helminths, which are worms, and helminths is a good word to know. They're macroscopic. They're all multicellular. If you're going to be large, you need to have multiple cells. And sort of like us, they have differentiated tissues and complex organs. And an example of a helminth that's not infectious are the earthworms that you see outside in your garden. They're quite closely related to some of the worms that infect people. Other type of parasites are much smaller. They're microscopic, so you can see them under your garden variety microscope. They're 2 to 10, 100 microns. And they're single cell. Some of them are amoeba. And the thing is, because they're small, they actually can live inside our cells or outside of our cells. So that's an important concept too, is just like viruses live inside cells and some bacteria live inside cells, protozoa, also being small but bigger than bugs, can live inside our cells and they can live extracellularly as well. So what you need for the objectives for this parasitic disease lecture is you must be able to describe, and not in gross detail. Okay, the questions I ask are very sort of general questions. You don't need to know life cycles in detail. You need to know what organisms look like and where they infect you, and how you can diagnose them. So part and parcel of that is understanding the life cycle of our favorite worm, which is Ascaris lambrocoides. The geographical distribution of parasites, and that's important because parasites are not all over the world in a general way, but they're located in certain places, and we'll talk about that. From a biological viewpoint, we need to compare the properties of intracellular and extracellular parasites and describe how that affects the disease. And the sort of poster child for single cell protozoan parasites will be the amoeba and Amoeba histolytica. So we'll talk about that. We'll talk a little bit about Plasmodium, which causes malaria, because that is very well studied at the cell biology, the molecular, and the epidemiological level. And because Plasmodium, malaria, kills still a lot of people in this world, we're going to talk a little bit about the treatment. Okay, so these foci, if you will, are on specific organisms, but what I want you to get is also a general impression of parasitic diseases, how they occur, and how we address them. So I've just told you the importance of amoeba, and this slide doesn't have amoeba on it, but what this slide, purpose of this slide is to show you that millions of people, you look here, millions of people infected. So this is 100 million. This is 1,000 million. It's a lot of people are infected worldwide with parasitic organisms. And parasites basically give nothing to us. They eat our food, they drink our blood, they reproduce in our bodies. Their goal in life is to keep living. It would be bad for them if they killed us immediately, because then they wouldn't have a host. So what parasitic infections generally do is they slowly sap us of our strength. Just taking enough nutrients to keep themselves going, but not enough to kill us, but certainly enough to make us feel pretty crummy and not be very effective at whatever we want to do. So remember the two groups, the helminths, those are the multi-cell organisms. And there's different types. There's flukes. We'll talk about those. There's tapeworms. You've all heard of tapeworms. And then there's the group of roundworms, Ascaris being one of those. And the roundworms are the ones that look very much like in their physiology to the worms that you see in the garden. Okay, they're long round tubes. Of the protozoa, I told you there's Plasmodium, which causes malaria, many different Plasmodium species. Leishmania, which we'll mention on, and then my favorite, of course, because this is what I work on, are the trypanosomatids. And you can see by looking here on the right that a lot of people in this world are infected with ascaris. Ascariasis, and it's about 10% of the world population has ascaris. These diseases are not so prevalent in the United States for a couple of reasons. One is we put screens on our windows to control the parasites that come via mosquitoes. The second thing is we break the fecal oral route of transmission by having flush toilets and what we call sanitary disposal of feces. And those are sort of two of the main reasons that parasitic diseases are not so prevalent in the United States, but in developing countries and that's it. So this is simply another way of looking at important parasitic infections. And that is by looking at a human body and looking at the different ways parasites can get inside of us. Parasites have been around for just as long as we have. So they've developed really clever ways of getting into our bodies. And some of them you've heard of, and some you might not have even thought about very much. So I like to think about acanthamoeba, which gets into your eyes. And I said just the other day to my husband when I was digging in the dirt and it was sort of spewing around and it was muddy, I said, Philip, this is a wonderful way for me to get acanthamoeba in my eyes. And he looked at me and said, what? So, well, yeah, acanthamoeba is a protozoan parasite that lives in the soil and it's around here and you can get acanthamoeba infection in your eye. Those of you who wear contacts, you might think about acanthamoeba because it's easy to get acanthamoeba infections in your eye if you don't take good care of your contact lenses. Because as I said, there's acanthamoeba around all over the place. Okay. So keep your glasses clean and don't let dirt go flying into your eyes. You can, of course, inhale through your mouth certain parasites, and it doesn't matter if these are protozoa or helminths. Your body cavities are there for all of these. So we have acanthamoeba, okay, Naegleria. That's something that about 1 or 2 people in the United States die of every year. If you go swimming in some water where there's Naegleria, it's usually swimming in a place where it's warm. It's a lake or a stream where the water's sort of warm. These Naegleria amoeba hang out. They can go right into your nose and then they go right to your brain. And there's not much we can do about it. So that's a problem. And there's usually about 2 cases of Naegleria a year. And they're usually in Texas or South Carolina, where it's pretty warm. Enterobius is pinworm. We'll talk about that. That's pretty common around here. So those you can get by sticking your hands in your mouth, or by inhaling rather through your nose. You can stick your hands in your mouth, fecal oral transmission, and get, as you see, a whole slew of different organisms. Everything from the worms, ascaris, to amoeba, to toxoplasm, to giardia, to our friend the pinworm, Enterobius, to cryptosporidium. And I haven't made this lecture where it's not a lecture of, it's not a parade of organisms. I focused on just a couple. I tell you that the CDC has wonderful websites for lots of these. I think one good thing that's come out of this terrible, 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 terrible pandemic is that we're all more comfortable, I think, looking at the CDC websites. And I think the CDC has actually improved some of their websites for all of us at all levels of education and backgrounds to get information. So I strongly support the CDC website, and it's chock full of interesting and important information about parasitic diseases as well as viruses and bacteria. Sexual contact, there's something called Trichomonas vaginalis. Makes sense, it's passed from penis to vagina. And that is something that we see in the United States. I was talking to a physician friend of mine who sees a lot of it in her clinic. Actually, this was in Texas, not in New Jersey, but that happens to be where she was. And she sees a lot of trichomonas going back and forth. Penetrate your skin. Okay. The hookworms penetrate through your skin. It's part of the reason, at least my parents and my grandparents always told me to wear my shoes. Because until fairly recently, we have hookworm. Actually, we still do have hookworm in the United States. And if you're walking around on grass where there's hookworm larvae, or you're walking around on sand where there's hookworm larvae or hookworm eggs, those little larvae will burrow through your skin and get into your body. And then finally, of course, our favorite, maybe least favorite, are the vector-borne. We have kissing bugs that pass Trypanosome cruzi. We have mosquitoes that don't only pass viruses and bacteria, but they pass parasites, okay, worms such as Wischiaria, that's elephantiasis, and Plasmodium, sandflies, and tsetse flies. So there's lots of different types of vectors that will carry, that basically work as syringes to carry parasitic organisms from the vector into our body via a jab, via injection. So these terms, move this, you might have heard a little bit more of this past year when we've talked about, when you've heard discussions about SARS-CoV-2 and reservoirs and bats and that. And so the term reservoir is sort of familiar to you all right now. And that's basically where a parasite or organism is in the environment. It often doesn't directly participate in transmission. That's not quite true. But the most important thing about a reservoir is parasites or the infectious organism hangs out there, but doesn't seem to harm the host. Bats don't seem to be harmed by SARS-CoV-2. Okay, the armadillo is a reservoir for Trypanosome cruzi, and it's not harmed by the trypanosome. And then vectors also are not so terribly harmed, but it's another useful term. And I like to think of it as the living transmitters. It's usually arthropods, right, which are mosquitoes and bugs, okay? And they can pass an organism on to us. An example that you probably learned about is Lyme disease, right? You get a Lyme disease from a tick, okay? You get West Nile from the bite of a mosquito. Well, you certainly get elephantiasis or Trypanosoma brucei, sleeping sickness, by the bite of a tsetse fly. And so it sort of makes sense that if the vectors are not in your area, then an infection cycle can't continue. Part of the reason we don't have malaria in the United States right now is because, as I mentioned, we put screens on our windows. So the mosquitoes don't bite us all that often. So let's do sort of a discussion now about helminths. As I said, worms, and there's different types. Excuse me. The worms are, I'll show you examples of these that I have in formaldehyde jars in a little bit. Okay, so stay tuned for that. They have to live mostly in the soil, so they have tough cuticles. They have a tough surface. Look at how giant Ascaris lambrocoides is. This is a female. There's females and males, just like in lots of other multicellular organisms, okay? And female and male are about the same size, but she's 35 centimeters long. That's about this long. And you can't see my hands, can you? Can you see my hands? No, you can just see the slides, right?
No, we can see it.
You can see my hands?
Yep.
You can see that 35 centimeters is about that long. Good. Okay. So those are pretty big. A tube, they eat at one end and produce eggs out the other. And flatworms look like ribbons. Okay. And here you see one really long flatworm. And if you look carefully, you can sort of see that it's flat. I like to think of it as those hair ribbons that we sometimes wear that have little striations in them. They're called gross grain ribbon. Maybe some people know about that. And they're much more primitive than roundworms. They lack a digestive tract, so they absorb food through their sides. And then finally, there's adorable little flukes or trematodes. And we'll talk about the most famous, which are schistosomes. And what you're looking at here, or that you were looking at there, is here's a fluke. Okay, it's a whole ugly sort of thing. It has a sucker on one end, hangs onto your intestine, or onto your bladder, or onto your liver. And we'll talk about the schistosome flukes in a minute. So as I mentioned, they reproduce sexually. You need both male and female. Okay, fine. Tapeworms, that's not the case, but that's a minor point. And they go through lots of developmental stages. And what's so bizarre to me is that these parasitic organisms not only have been around for so long, but you'd think they would have streamlined their life cycle, and they haven't. They have life cycles that are far more complex than ours are. And who knows why? There's a lot of fun books to read about this. One of them that I highly recommend that's fun to read, called Parasite Rex. It's by Carl Zimmer. He still writes for the New York Times. I always make sure I read Carl Zimmer's articles in the Times. And this book called Parasite Rex, which I can lend you a copy of if you can't find it on Amazon, is fun to read because it goes through the kind of the weirdness of parasitic infections. So here I have developmental stages. Some must occur in the insect or animal reservoir. And therefore, the point of the fact that there's developmental stages means that it's often not human to human transmission, okay? Unlike in the case of SARS-CoV-2, the virus, right, where there is human to human transmission, there's not that many helminths that are easily transmitted human to human, okay? Unless, of course, you pick up eggs that are lying on the ground in dirt and stick your hand in your mouth. Any questions so far? Okay, Ascaris. I told you, so this is an old-fashioned, it's a regular ruler. Look how big these guys are. Okay, a quarter of the world's population is infected with Ascaris. And that's because the Ascaris eggs, the female Ascaris produces hundreds and hundreds and hundreds of eggs every day, and they end up on the soil. You have Ascaris in your gut. The eggs are produced in your gut. They're pooped out with fecal material. They sit in the soil, waiting for the next person to touch that soil and then put their hands in their mouth. I said in the US, it's largely confined to the rural South because where there's inadequate sanitation, and I'm not saying the rural South, I'm not making any political statement here. I'm just saying that in any area that's poor, where there isn't a lot of sanitation, it can be your neighborhood, it can be any neighborhood, okay, any environment, that's where you're going to find more Ascaris worms. The thing about having Ascaris is it's not really that bad. If the Ascaris worms are living in your intestine, they eat, you eat, you're fine. Problem is when you get a whole lot of Ascaris worms in your gut. And imagine just having in your belly all of these Ascaris worms. For every egg you accidentally consumed, you have a grown female or male Ascaris. So that's the problem with having Ascaris in your body. They live in the small intestine. They feed on semi-digested contents. That makes sense. Lots and lots of eggs are laid every year. And the eggs, if you're going to be an Ascaris worm and you were going to design the eggs that you're going to produce, you'd have a lot of these traits in your design. You'd want a thick shell. You'd want a larva that stays inside the eggs while it's protected. You want your eggs not to hatch until they're ingested into someone's gut. And then finally, maybe just for fun, you'd want this incredibly ridiculous, complex life cycle that the adults go through, which I'll show you on the next slide. Okay, this is actually bizarre, okay? This is a slide taken from the CDC to emphasize what I'm telling you, which is that the CDC slides and pictures are really good. Okay, here's an Asker's egg, okay, in fecal material. The way you can, fecal material consists of several things. Most of the stuff in your feces looks like cut up celery or chopped up celery, sort of this stuff, right? And this bacteria, chopped up stuff. Then when you see something that's large like this, it's an Asker's egg. And this is a little deceiving because most eggs from parasites have a smooth surface. Asker's doesn't. So why am I showing you that one first? Well, we're starting with the most difficult one, but it's a nice big thing. Here's the egg shell, its bumps, and then you see inside the developing embryo. And here, this is even a better picture. Here's the egg shell and the developing embryo inside. Really sort of cool to see. So let's go through the life cycle. Let's start with this stage, okay? Even though that's not how this picture numbers them, I'm going to start with the transition from 3 to 1, from 3 to 4. So here's an egg with its bumpy surface, and you accidentally stick your hand in your mouth. Why might you do that? Well, let's say you're someplace where there's an orange grove, okay? And there's an orange that's fallen on the ground. That's fine. You pick it up and you think about eating it. Okay, you're not going to eat the skin of the orange. However, if that area, if that orchard has been fertilized with what's called night soil or human manure or human feces, then it's very likely that the orange on its surface is going to have Asker's eggs. So you peel the orange, you still have eggs on your fingers, and now you eat the flesh of the orange, but your fingers have Asker's eggs on them. So the Asker's ends up going into your mouth, okay, into your mouth, down into your gut. So we'll follow 4 to 5. And then the coolest thing, well, it's awful, but happens is that the larvae come out of the eggs and they go through this complex migration just to end up back in your gut. They leave your intestine, they go up into your lungs. Okay, these are the little larvae. And sometimes you feel like you're coughing. It goes up your trachea, and then you swallow them down your esophagus. So you can see worms coming out of people's noses. Well, the worms are going through this cycle, okay? Down to your esophagus, down to your gut, and that's where they hang out, male and female, producing eggs. And I don't care about fertilized or unfertilized eggs. That's not the point of this. The point of this is they go through all this complex stuff in their life cycle. And here's a picture of the worms. As I mentioned, if there's a couple of worms in your gut, you're fine. If the worm load is heavy, you don't feel well. And if the worms decide that they don't like sitting in your gut and they break out of your gut and they go to your liver or someplace else, into another organ, then you get really sick. So if you travel to some wonderful destination, okay, where you might have picked up Ascaris, you should be somewhat concerned, because even if you're not terribly symptomatic, even if you just are concerned that you might have in fact gotten an infection, or maybe you've gotten another type of infection and you want your physician to look a little more globally, you should think about looking for Ascaris eggs in poop, in fecal material. You can even do that yourself in the lab. So here you see the awful, awful picture of this young boy, okay, with an Ascaris worm, okay, coming out of his nose and his mouth. And here you see a patient who's been treated with a vermifuge. A vermifuge is something that stops the verms. It's called albendazole, and it blocks the tubulin specifically of the Ascaris worms. The worm stops moving around so much, and then you can poop out the Ascaris worms. So look at how many worms have just come out of this one kid's anus. Disease you don't want to get. So we're going to talk a little bit about flukes. I think these guys are very cool. They're called schistosomes. That's the main type of flukes we think about. There's different species. Mansoni, Hematobium. You think about schistosomes, what's their claim to fame is that they have to go before, when they go from me to you, they have to go through a snail intermediate. And that snail lives in water. So if you think about areas where there's been water that's been dammed up, especially in warm areas where there's large numbers of snails, a special kind of snail, then you shouldn't go into the water because of schistosomiasis. And actually there's areas in poorer areas of Southeastern Asia and in Africa, and actually in general areas of Asia where schistosomiasis is actually fairly common. And often a young boy, by the time he's 13 or 14, is going to start have bloody urine because he's got blood flukes that have been hanging out and sitting in his bladder, okay, and causing some bleeding into the bladder. It's also called bilharzia. Many of these diseases have very interesting regional names, and bilharzia is the one that's used in Africa. It's second only to malaria as its most devastating parasite. You see here the females and males. These guys are just adorable. Here's the male, and here's the female, and they actually hook up together for life. They mate for life, and their job in life is to produce eggs. And that's actually the pathology, because those eggs get stuck. You make an immune response against those eggs. You make granulomas around those eggs. And if you have a lot of schistosome eggs in your liver and a lot of these hard masses called granulomas, you get portal hypertension, your belly swells, and you eventually die. That's the problem of long-term schistosomiasis. These are the areas in the world where you see schistosomes. Okay, certainly when the Aswad Dam was built, now 50 years ago, there was an increase in schistosomes and schistosomiasis there. You see it in these parts of the world and this part. This is where the snails are. And here's what happens. You poop or pee out the eggs. The eggs are not quite as interesting as the Ascaris eggs, but I think they're sort of neat looking. They're oblong, and they usually have a point on them. Sometimes the point's on the side, sort of looks like a keel on a boat, on a sailboat. And these eggs have to go into a snail to go through developmental steps. And after they do that, they come out of the snails as little cercaria. I think they look like little torpedoes. And those penetrate your skin when you're in the water, bathing, washing, playing around. And once that happens, the schistosomes get into your circulation, and they can either sit in your liver, your intestine, your bladder. And then, of course, like with Ascaris, produce eggs. Eggs come out and keep the cycle going. For a lot of these parasites, it's all strength in numbers. Okay, the more eggs they produce, the more they are assured of their longevity and their continued life. So look at some of these eggs, really. So here's an egg. Okay, this one has a nice smooth shell. You can even sort of see the little embryo inside, and you see this point. I like to think of this point as what's really sort of causing the problem, at least in hematobium, okay, that's poking holes in the sides of your bladder and causing bloody urine. We have treatments for most of these parasitic infections, as you can imagine. Treatments are drug companies in all of the world do not make much money from providing and from developing drugs against diseases of the poor. And so a lot of our drugs are either old drugs that have been around for a long time, or things that people have taken off the shelf that are used for other infections. We use for schistosomiasis, we use praziquantel, and it works pretty well. Obviously, a way of preventing this spread of schistosomes would be to get rid of snails. But as you have heard in your lifetime, when you upset an ecological balance, that can be a problem, right? If you get fish that eat all the snails, and you're going to have too many fish and you're not going to have the right kind of predators, blah, blah, blah. So upsetting the biological balance in the world is something that we've seen when we've tried to control schistosomiasis in one case, okay, by eliminating snails, by having fish eating snails. All right, so now we're going to look at protozoa. So we've gone from worms that can either be sort of little, like the schistosomes, okay, that hook up together, or the ascaris worms that are large. Now we're going to look at protozoa, which are smaller, okay, single cell, again with a nucleus, okay, but only a single cell. And your favorite, or my favorite, well, I guess the favorite of biology teachers in 8th grade is the amoeba that you look at when you look at pond water in 8th grade biology, okay, or maybe nowadays 6th grade biology. So here you see an amoeba. It's sort of schmoozing around. These pseudopodia come out when it's looking for food. It's eaten some food. You can see that in here. Okay, they actually eat red blood cells. Yum, yum. Remember, eating red blood cells is a really good thing, because by eating red blood cells, you get a lot of nutrition. So parasites like to hang out in your gut where there's a lot of food, or in your bloodstream where there's also a lot of food, which are both rich in nutrients. Some of them don't schmooze around. Some of them have flagella. It's an adorable guy called Giardia lambia. Here's a real one, and here's a drawing. But if you look at this Giardia, remember it's single cell, it has these suckers that look like 2 eyes, and those hook onto your intestine. You've probably heard of giardiasis, because if you get traveler's diarrhea, it's very often, it's sometimes bacterial, but it's very often due to giardia or giardiasis. So if you come back from a trip and you have very bad smelling poop and lots of cramps, it's very possible there's giardia in your system. We can treat it, but it's called the monkey face because it sort of looks like a monkey and it's got lots of flagella. We're not going to talk so much about ciliates, but there are forms that move around not at all. And this would be, for example, a gamete from a plasmodium. So the purpose of showing you this is a single cell sexual stage of malaria, simply. Okay, so protozoa need to move in various ways. They can have cytoplasm projections, they can have flagella, or they can just sort of hang out, or they could have cilia that beat. Purpose of this is to get around and to find food. We often group the protozoa by disease, okay? And that's because, as I said, parasites have been around for a long time, way before we had DNA sequencing. So most of the names for parasites and the descriptions and the way we categorize them is not so much based on their gene sequence, but based on where they are in the body, in the human body, okay? It was a medical field before it was a microbiology field. So you can have them in the intestines or the urogenital tract, blood or tissue. They can be intracellular. It's good if you an intercellular parasite because, as I said, you can avoid the host immune response. You can hide out inside a cell. You can eat that cell. What you can't do, though, is be tolerant to desiccation, whereas the extracellular ones, they don't make eggs. This is an important distinction. Single cell organisms don't make eggs. They make cysts. And what that means is you have a feeding form called the trophozoite. And when things are not good, and when they're stressed, or there isn't a lot of food around, they very often simply form a shell around their body or a layer of protein and lipid and sugars, and that forms a cyst. So this is not the cysts that you see on your foot or on your finger, or we talk about a cyst in your brain. The word cyst here simply means a single cell organism, okay, that's really stable and really dormant, okay, because it's in a cyst form. Sort of like you learned about spores for bacteria. Well, amoeba have cysts. Intestinal infections, amoeba and giardia, we mentioned those, and urogenital trichomonas vaginalis. And these guys are just adorable. They sort of look like little leaves and they sort of float through vaginal secretions. So here's an amoeba. Okay, it's actually eating a fibroblast. They'll eat anything. Okay, so this whole thing is the amoeba, and it's chomping away. It's sucked in. Okay, by putting its pseudopodia around it, this fibroblast. The fact that amoeba can eat just about all of our cell types is why we get tissue destruction. They'll eat through your colon. It's not good. So let's look at an amoeba. When you fix the amoeba, they don't look quite as interesting as I showed you in the first slide. Okay, when you fix them with formaldehyde, they sort of round up. So here's the amoeba. These are stained, so the nucleus, the nuclear material is stained, and you can see that. And then this other material here is basically the cytoplasm, sort of nondescript. A lot goes on, but sort of nondescript. In contrast, you see a cyst, which is rounder. You can imagine if you were dormant, you would want to sit around and be a round thing. You're not going to have pseudopodia that go out and look for food. And you're going to have a nuclei inside, and you're going to have sort of some other structures. Don't worry about those, okay? And you can see this in this fecal sample. You can see these bits that look like broken celery and the nice cyst, okay? So here's a cyst with several nuclei. Okay, there can be more than one nucleus in a cyst. Okay, when it gets back into a person's gut or conditions are better, it excists, and certainly my colleagues in the molecular biology field, the molecular genetics, study the genetics and the molecular genetics of how excistation happens. A whole new genetic program is turned on. The organism gets rid of its wall, and it starts looking like what's called a trophozoite, which is the feeding form. And now it can go around and invade your tissues, and go into your liver, or it can just hang out in your gut. And I have so many of the, so a little bit more details, again, using the CDC slide, okay? Sort of in and out, simple life cycle. Okay, entamoeba cysts sit on the ground, causes diarrhea. And people are really the only reservoir for entamoeba histolytica. There's certainly lots of other types of entamoeba, but we're it for entamoeba histolytica. And here's sort of a nice example of how the organisms on this drawing can invade your tissue. So here you have, think about this, this here is the lumen of your gut, okay? So this is the surface of your gut right here. And the amoeba are sort of hanging around on your gut, all fine and good. You excrete the cysts, not too terrible. You have diarrhea because you've lost some of the surface of your intestine for absorption. But the amoeba can also have ways that they can sort of eat through the mucosal layer. So you see these amoeba here eating through, they have little nuclei here, eating through. And when they do get into your system and you have an invasive infection, okay, that's when the problem starts. So again, you want to really, in your own lives, and in lives of your friends, if someone has a diarrheal infection, you really want to check it out and see what's causing it. And let's look at this slide, which is a real example of what I was telling you. So here's the mucosa membrane. Okay, that's there, it's intact. It's lost there, and it's intact there again. And you see the amoeba have eaten into this piece of intestine. Okay, not very good. Okay, they're ulcers. So the amoeba have destroyed the nice surface mucosa and eaten into the tissue. Not good. I don't think. We'll talk a tiny bit about host defense. Mainly, it's innate immunity that we use to protect ourselves from these infections. This simply just, the point here is just that there's proteins on the surface of the amoeba. It's often that instead of just looking at fecal smears to diagnose intestinal parasites, we use ELISA assays, and those ELISA assays are based on antigen-antibody interactions, and the antigens are those that are found on the surface of the amoeba in this particular case. Lectins are nice and sticky, so amoeba exploited lectins to put on their surface to help them adhere to your gut so they can happily munch away and reproduce by binary fission until times get bad and they insist, okay? So I didn't mention that, but protozoa reproduce by binary fission. Okay, sort of like when you learned about paramecium when you were younger. Simply another discussion of amoebiasis. This is, as I just showed you, really, here's the amoeba, okay, sitting on the mucus layer. So you can choose whichever picture you like better, either the real ones or these colored diagrams or the black and white diagrams. And then the enta-amoeba will basically pass through the mucosa. Okay, there's a certain amount of inflammation that happens, as you can imagine. And you get invasion. So you don't want your amoeba going into any place else except your gut. And I've kept this slide in. I'm not quite sure of how to explain it. This is from Dr. Garrett. I think its point, maybe Ian can help me, maybe someone can help me understand this better. It's this guy looking obviously at this mammoth and seeing that it's a mammoth, even though he's only looking at a tiny piece of it. And so what maybe the way to interpret this is that very often parasitic organisms hide. And so you have to look carefully and you have to look at sort of the whole picture of the person in order to be able to understand what's going on, okay? If you look, for example, for pinworms in feces, you won't find them. You have to look for pinworms specifically by looking at the perianal area of a person, okay? So I've probably beaten that up enough. But the point I did mention is that there are specific antibodies that are not only helpful for invasive amoebiasis, but they're also very helpful for ELISA tests, which is the serological test that we generally use for diagnosis these days. Treatment, pretty straightforward. It's nice. It's metronidazole and incorporates into DNA. And you prevent infection by boiling water okay, by good sanitation and by using iodine. So this is sort of the basic things that you learn about when you worry about a breach in a water supply, right? You boil it, you filter it, okay, for all kinds of infections. Giardia I think about a lot when I go camping, because giardiasis is around in all of our streams, okay, and waterways. And the reason why you really don't want to be eating out of a stream like this little kid is because you'll get giardia and you'll end up with a bad GI infection. I showed you pictures of giardia, Giardia lamblia. It's a flagellated protozoa. You get really awful acute watery diarrhea and dehydration when you have giardiasis. It's fecal contaminated water. And what we often talk about is and beavers, remember we talked about reservoirs, beavers are actually the reservoir, as I show here, for giardia. So if there's a beaver dam up the stream and you're downstream camping and you want to drink the water, I really wouldn't do that. I would filter it first or treat it with iodine or bring your own water or boil it, because giardia is there and it'll get into your gut and stick to your gut. See here, contamination of water, food, blah, 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 blah. It looks like a monkey face, I told you. Very, very primitive guys. They have a cyst wall. Okay, and you don't want them stuck in your gut. And this is great. Here's a piece of gut. Okay, you can barely see the mucosal surface here, but each of these is a little giardia guy. See, they're sort of curved shapes so they can hook onto your gut. So here's 1, 2, 3, 4, 5, 6. And these stainings that we do, this Giemsa stain or H&E staining, you get a very dark staining of nuclei of cells, and you get a lighter pink staining for cytoplasm. And on something as small as giardia, you almost can't distinguish in this picture the nuclei from the cytoplasm. Okay, the final one we're going to do before I do our show and tell is we're going to talk about Plasmodium, because we've all been, many people have worked for many years to develop a malarial vaccine. We're not quite there yet. As you might know, the Gates Foundation is focused not just on tuberculosis, but on malaria as well. Malaria kills millions of people every year. Excuse me. And it's an infection of the blood. We'll talk about that. We won't talk about toxoplasm, another protozoa infection of blood and tissue, trypanosomes, and leishmania. Now, let's look at malaria, okay? So you get bitten, you get infected with malaria only by the bite of a female Anopheles mosquito. So you have to have Anopheles mosquitoes, you have to have females, and they have to be interested in taking a blood meal so they can produce eggs. So here's a mosquito, an Anopheles mosquito with its gut, its body full of blood from a person. I'm sorry for the ringing. This is sort of a nice picture, but we'll look at further ones of the life cycle of malaria. Let's go right to this slide. Remember I mentioned at the beginning, there's these incredibly complex life cycles of most of these parasites. Well, I think Plasmodium takes the cake for this, okay? And we're just going to go through a little of that just so you appreciate the complexity, number one, and so you can understand why it's so easy to increase the amount of drug-resistant Plasmodium in our world. And we've really accomplished that pretty effectively. Most of the Plasmodium in the world is chloroquine resistant. Chloroquine you might have heard of. It's derived from quinine. And quinine is a drink, a liquid you can drink. It's made with cinchona bark, and it's been known for a long, long time that drinking quinine, okay, will ameliorate the symptoms of malaria because of the chemical agents in the bark. And so because it does that, quinine is often called tonic. So some of you might have heard of gin and tonics, and it's really gin and quinine. And so that's sort of a long explanation of why quinine and tonic, why quinine is called tonic. Okay, so let's go through this. You get bitten by a mosquito. These sporozoites, so they're single cell, remember, they're actually long and thin. They have nuclei, that's what the dots always are. They go right to your liver, and the purpose of going to your liver from your bloodstream is so they can increase in numbers. Go into your liver. A few sporozoites actually emerge from your liver a couple weeks later, depending on the species, as these little round forms. And then they go through what's called the asexual cycle, where they spend their life infecting your red blood cells, growing inside your red blood cells. They're sort of funny. They look like rings inside your red blood cells. And guess what? They're called ring stages. And inside the red cells, the rings develop. They become feeding forms, trophozoites. They then fractionate into little things called merozoites. You don't need to know these words, but I can just tell you that an infected red cell looks like a pomegranate, with each of the seeds being one of the little merozoites. And the point is that when they break out of the red cells, that's when you get this exacerbation of fever, okay? Because this cycle happens after a couple of days in a very synchronous way, which is why you probably know malaria is characterized by these cycles, pretty periodic, depending on the species, of fever and chills. You get fever when the parasites all break out, the merozoites break out of red cells, and then chills, and then the cycle continues. It's almost hard for mosquitoes to get Plasmodium because some of the parasites have to go through the stage called gametocytes. And the reason why this is interesting is, as you can imagine, there's so much differential gene expression that happens in Plasmodium, as well as in trypanosomes, which is what drew me to the study of trypanosomes. Anyhow, these gametes go into the mosquito. Lots of biology happens in the mosquito. You think mosquito would be just a vector, just a syringe, but it's not. And it's actually in the mosquito, okay, where mating between these gametocytes happens. So if you happen to have a drug-resistant gametocyte or gamete and a drug-sensitive one, then after they mate and produce sporozoites, all the sporozoites now are resistant to drug. So the fact that there's this mating, okay, of these single cell organisms, okay, of these gametocytes, and just genetic shuffling enables, or sadly enables, drug-resistant Plasmodium to spread. So again, the mosquito is not just a syringe, but it's an active environment for Plasmodium life cycle changes. So this is sort of cool. You might have seen this picture before. This is a false colored scanning EM. So here you're seeing a red cell, and here you're seeing a red cell, and here you're seeing a not very happy red cell. It's got some merozoites breaking out of it. And this is even more dramatic. Okay, here's a red cell that's broken open with a little merozoite hanging on. And here you see in less graphic color, a red cell with a little merozoite trying to get in. Great place to live inside a red cell. Lots of food, protected from the immune system. Couldn't be better. And as many of you probably know, there are regions of the world that have been so inflicted with malaria that many of us have developed red blood cell and hemoglobin genes that make our red cells a lot less tasty and a lot less easily to be invaded by malaria. Sickle cell is one example, and another are the beta thalassemias. And in fact, my family is from southern Italy, and in my father's side of my family, we carry a beta thalassemia gene. I'm a heterozygote. My son's a heterozygote. And that gene has been passed down because long, long ago, people in southern Italy, where there was a lot of malaria, those people were protected if they had a beta thal trait. Just like if you have a sickle cell trait, okay, you're protected to a certain degree from malaria. So Plasmodium has been around so long that humans have actually evolved with it. The outcomes, fever, we talked about those. It's really terrible, but you can die from some Plasmodium infections. And we'll talk about that just briefly to wrap up. So here's a blood vessel. Okay, these are the cells that are lining the blood vessel. Okay, here's an infected red cell. And when red cells are infected with Plasmodium, the Plasmodium sort of throws this anchor out to the surface of the red cell to hook the red cell onto the capillary. If the red cell gets taken to the spleen, your spleen is going to see it as infected. It's going to kill the infected red cell. And Plasmodium is not going to live. It doesn't make Plasmodium happy. So it attaches itself, at least only in the case of falciparum, the most deadly of Plasmodium infections. It attaches itself by generating, transcribing, translating, and then moving one of its proteins to the surface. And those proteins happen to be called Plasmodium falciparum, EPMs or EPMs. And the point I want to make is simply that the red cells will then stick to the surface. Okay. The surface protein is necessary for the cytoadherence. And for the cells to stick to each other. In fact, that's what happens. This is why you die, in fact, is because you get capillaries in your brain in falciparum that are clogged with infected red cells. And here you see an infected red cell with healthy red cells hanging on. As you can imagine, you wouldn't want that anchor to always be the same protein, because if it was, you'd make an immune response against that and the Plasmodium would have nowhere to go. So in fact, Plasmodium changed these EMP proteins, okay, in something called antigenic variation, which is a term you might have heard in the case of, you might have heard antigenic variation when you've heard about Neisseria, okay? Lots of small organisms change their surface coats in ways that they've developed to basically avoid the host immune system. So there's a large number of these different surface proteins, and what you can see in Plasmodium week after infection, for example, here, you see all the Plasmodium have one surface type and then a different surface type and a different surface type. And again, these surfaces types, these proteins are on the outside of the red cell. I've just told you all about that. I mentioned like Neisseria. It doesn't involve DNA rearrangements. It involves epigenetic reactions. And that's something that many of my colleagues have spent their life studying. Diagnosis, large parts of the world still have malaria. You can do a blood stain. Giemsa is just a cheap and inexpensive stain you can use. You stain the nuclei purple and the cytoplasm pink. And where there's mosquitoes, you see. Malaria. What's really used a lot still are bed nets. If you sleep under a bed net, then the mosquitoes can't get to you at night and infect you. Bed nets are a wonderful, cheap way of protecting us against malaria. We also use now in the United States and in Europe, finally, a derivative of a plant that's been used in China for millennia called artemisinin. And I remember the first time I saw the artemisinin plant, I thought that was just so absolutely cool. But it's derived from the artemisinin plant. An extract is made that helps treat malaria. Okay, chloroquine I mentioned and quinine. We don't have a vaccine available yet. There is a candidate in phase 3 studies. It's called RTSS. Okay, it's a chemical. And the reason I'm bringing it up is not that you need to know it, but you need to know that people are working hard on vaccine candidates for malaria. Certainly when I gave this lecture last year, we weren't even thinking so much about vaccines weren't in the newspaper every day. Now they are because of SARS-CoV-2. And so I think the entire world has a better appreciation of the importance of vaccines. And we all certainly hope that we have a malaria vaccine at a reasonably soon time. And that's what I want to show you. I can stop sharing my screen. Okay. And now, how can I make myself look larger? Can someone tell me?
You're all set.
What?
You're
Say again?
fine. See you.
You can see me in a large format. I just can't see me in a large.
Yeah,
format.
everybody individually can do this, Vivian. They can just, change their view to see you.
So I could change my view to see me. Okay, now I can see everybody. This is cool. All right. All right, so guys, if you can't see this well, tell me, speak up and I'll show you. Okay, but let's start with the fun one. What is this? My son gave me this for Christmas. Can someone tell me what it is? Please?
Mosquito.
The mosquito. And look what this mosquito has in it. This is just so cool. You can unzip this mosquito. This was the best Christmas present. And you can see a sporozoite. This is the form of malaria that gets into your body when the mosquito bites you. But as you know, mosquitoes carry other things, thus the other things. And we have here dengue fever, virus. This is so cute. We have Zika. And we have, let's see, West Nile, dengue, and one more. Dengue and West Nile. So isn't that cool? Okay. And then you can just zip them all back up into the mosquito. I just thought that was a wonderful, wonderful gift.
That's awesome.
Isn't that cool?
Yes.
This is what happens when your children grow up with a scientific parent.
When they finally
I also-
figure out what your job is.
Yeah, exactly. Okay, so this is another. These are how, can you see this? Talk to me, people.
Yeah, I can actually see that. I can see that
Yes.
pretty good. Yeah.
Okay, those are Ascaris worms in a jar. These are taken from one kid, one young individual, one young person from the hospital here at University Hospital a bunch of years ago. Okay, so there's lots of these worms. And everyone, you're welcome to come see these in person in my office in the ICPH building. You have my contact information. I'm here most days. Give me a holler. Okay, you can come see them. Let me show you some more things. You
Yeah, I
kind
can
of
see that. Yeah.
It's a tapeworm.
Huh.
Okay, there's a tapeworm. Sorry, got yet someone raising their hand, or is that me? Someone have their hand up? It's me. Okay. Tapeworms, come and see these. Another, this is a little cloudy, another wonderful tapeworm. This actually looks even better, doesn't it? Look at that. Isn't that great? Can I hear some oohs and ahs, please?
Oh my goodness.
Please? Oohs and
Oh.
ahs?
Ooh.
How about an ah? Ah, thank you. Very good. Thank you. Thank you. All right. Another asterisk. That's not so exciting. And that's basically. I have some malaria mosquito larvae. You know what mosquito larvae look like. So that's pretty much it. Please come to my office and see these in person. You can't touch them because they're in formaldehyde, but you can look at them up close and you can hold the jar yourself and you can photograph yourself with them if you'd like for your friends. And questions, please.
Are the worms normally stored that way, or do they get stored in the packaging that the malaria was in?
These worms are normally stored in formaldehyde. Okay.
Yeah.
Like this. So these are real. The ones that were in the mosquito are stuffed. Is that what you're asking, or am I not?
Before
understanding?
you had some boxes there, you said they were mosquito larvae.
Oh, yeah, I'll show you those. Those are embedded. Can't see this very well. You can't see this at all. Okay, they're
Right.
here.
I was just wondering why they were stored differently compared to each other.
Probably because these are so small. And so the company that made these little blocks, okay, did it this way. What's really cool here is that you can see also, I don't know, you guys have to come see this. You can't,
Try
The
putting
little
try putting a piece of paper behind it,
okay.
Vivian, maybe they'll, okay, now put it right to the screen. I think we can, oh yeah,
You
that's
see
better.
that thing? That's a bed bug, guys.
I
The thing
see.
in the middle is a bed bug. The thing up
Wow.
there is a mosquito. Look at the bed bug. Okay, and then the thing on the bottom here, it's a rat flea.
Interesting.
And here's the mosquito bits. Okay, the different stages. These are just sort of fun, right? I have a housefly too. Different stages of a housefly. They're just fun. You have to accept that we think these are fun. I think these are fun, okay?
All right, so if there's no more questions, thank you very much, everybody. Thank you, Dr. Bellafato. I'm going to stop the recording.